Biofilm, in essence, is a community of microbes growing on a surface. They are a great example of synergistic relationships between many microorganisms. Often, a biofilm is composed of multiple different species all living together, communicating and coordinating with one another, much in the same way cells and tissues in multicellular organisms do. Sometimes, organisms that would not normally be pathogens can become pathogens when in a biofilm. In fact, the Center for Disease Control and Prevention estimates that biofilms cause up to 70% of bacterial diseases in industrialized countries. Biofilm can attach to many surfaces such as teeth and the mucous membranes of the digestive system. Dental plaque on teeth are an example of harmful biofilm. These biofilm can erode teeth, leading to cavities. There, in general, are 6 steps to the full formation of a biofilm. The first step is when free living cells come in contact with a surface. The second step occurs when those cells attach to the surface. In fact, some cells have structures that help them attach to surfaces more readily, such as fimbriae and glycocalyces. And these structures seem to aid in the formation of biofilm by helping them attach to surfaces. The third step is developing an extracellular polymeric substance, also known as an EPS. This substance is a gooey matrix that has many important functions in forming a biofilm. It adheres cells together, sticks cells to their surface, forms microenvironments in the biofilm, sequesters nutrients, forms water channels in the biofilm between organism groups, and may protect the cells in the biofilm from stressful environments such as UV radiation, antimicrobial drugs, and changes such as pH, temperature, and humidity. The EPS serves a huge function in protecting the cells in the biofilm, likely allowing them to survive in harsher conditions than those cells would typically survive in, allowing the biofilm to persist in many different environments. For the fourth step, microbes in the biofilm secrete quorum-sensing molecules to relay information among members of the biofilm. Quorum sensing is a way for microorganisms to respond to the density of nearby organisms, such as in a developing biofilm. The way quorum sensing works for microbes in the biofilm is that microbes have receptors that can bind the secreted quorum sensing molecules, taking up information about the density of the microbes in the surrounding area. The molecules begin binding to the microbe receptors when the density of the microbes in the surrounding area surpasses a threshold, creating many microbes, which makes it much more likely for them to bind to receptors. When microbial density is low, it is very unlikely for molecules to bind as there are not as many molecules. Molecule density rises with microbial density. After microbes are bonded with the quorum-sensing molecules, it activates suppressed genes, changing the microbes to be better adapted for the biofilm, allowing the biofilm to strengthen. Ways in which these microbes can change include changing size and sexual ability. The fifth step, made possible by the quorum sensing process, is that members of the biofilm take on specific roles in the biofilm, much like how tissues in a multicellular organism have different functions. The sixth and last step is that some cells or groups of cells can leave the biofilm, possibly to start another biofilm elsewhere. The quorum sensing and the final step in biofilm formation are very important when it comes to biofilms interacting with immune systems. Generally, immune systems identify and annihilate invading pathogens. However, some pathogens can hide in the body while still few in number and only become visible to the immune system when quorum-sensing molecules show that there is a high density and biofilm, causing disease. At that point, it is difficult for the immune system to fight off the pathogens. If a biofilm is able to develop in one area of the body, the cells that are able to leave the biofilm could hide in other areas of the body long enough to multiply and form a new biofilm in that new area of the body, causing the potential spread of disease. Thankfully, researchers have identified multiple potential ways of controlling biofilms. They have discovered drugs which block microbial cell receptors, thus blocking communication between cells and inhibiting biofilm formation. There are also enzymes that degrade the EPS, allowing antimicrobial drugs to reach the cells in the biofilm that were previously protected by the EPS. Finally, they have experimented with artificially amplifying quorum-sensing molecules, tricking hidden cells into thinking the density of biofilm is high enough to reveal themselves to the immune system, allowing the immune system to destroy them before they multiply and become a disease in the body.